Good morning, everyone. My name is Jesus Antonio Sanchez-Perez. I am a PhD student at the Georgia Institute of Technology, and I am happy to present our work titled Initial Validation of Multi-Frequency Patch-Based Impedance Pneumography in Hospital Settings. This will be the outline for today's presentation. I'll first cover the motivation and firework. Then I'll go into the methodology that we followed in our paper. Then going into the results with comparison of feature trends and some exciting exemplary waveforms from the hospital. And finally, I will conclude with some thoughts on the impact of this work as well as the future work. So what is the motivation for this work? Some of you may know, particularly in the area of COVID-19, there are many conditions that are characterized by signs and symptoms of respiratory stress. And these include heart failure, acute respiratory stress syndrome, pneumonia, and many others. And these signs and symptoms are shortness of breath, labored breathing, fatigue, etc. Certainly, we can expect that respiratory markers such as respiratory rate and tidal volume and many others are going to change, and therefore it is important to measure them. However, they are mostly measured in advanced hospital settings when patients are receiving advanced forms of treatment, and often when they're measured somewhere else, it is done in an inconvenient way. And we have, for example, the gold standard spirometry, which consists of inserting this breathing tube that you have to breathe in through in order for it to capture the lung volume changes accurately. So what this tells us is that there is a need for accurate and convenient wearable respiratory sensing systems, and we think that impedance pneumography is a promising respiratory sensing modality to address some of these issues. And what is impedance pneumography? Briefly, it is a non-invasive bioimpedance technique that consists in its conventional form of using two electrodes, two pairs of electrodes, one pair to inject a current through the thorax, and another pair to measure the corresponding voltage drop in the thorax. And what that will give us is a complex impedance vector that the real part, which is the resistance, is strongly correlated with lung volume. And to illustrate that further, we have these figures on one of the early works in this area where they were comparing the spirometry gold standard volume signal on top, both in frequency and in time, to the IP signal below in frequency and in time, and we see that there's excellent agreement. Another great characteristic of IP, as with some of the, or most of the bioimpedance techniques, is that in the multi-frequency analysis, it could provide information about the underlying tissue composition because it is known that low frequencies excitation currents travel mainly through the extracellular spaces, where the highest frequencies travel mainly through, can travel through both because they can penetrate the membrane of the cells. So we can add some remarks on what are some of the key benefits of IP, as providing a continuous respiratory waveform in a non-invasive way. And this could allow, for example, for breath-by-breath feature calculations. It has excellent agreement with the spirometry gold standard. It could provide underlying tissue composition information through the multi-frequency analysis. And as you will see, there are many ways and many options for placing the electrodes and configuring them. And this is precisely where some of the recent work in this area has been focused on. We have, for example, starting from the left, the conventional tetrapolar IP. We can see the electrodes and the wires coming from the sides, from the mid-axillary lines to the sensing platform. These are recent work from our lab. And then we have the patch-based. These are more convenient ways of measuring IP, so the patch-based bipolar configurations. The placement is now simplified by using two electrodes rather than four. And then we have the patch-based tetrapolar IP, which is a work that we recently proposed. This is a work from my colleague, John Berkebile, that consists of this 5 centimeter squared electrode arrangement that's placed below the jugular notch. And that has been validated in both active and static conditions in a controlled environment, in a healthy population in our lab. And we were interested in seeing how this patch-based tetrapolar IP would perform in an uncontrolled environment, such as a clinical setting. And this is exactly what this paper is about. The way we conducted this initial validation was that we compared both the patch IP and the transthoracic IP, measured using our wearable multimodal sensing platform that measures multi-frequency IP, each sampled at 16 hertz. And in collaboration with the Grady Memorial Hospital in Atlanta and Emory University, we recruited 7 patients with heart failure and took measurements on admission and discharge. There were 5 of the 7 patients with 2-day measurements that we used to compare trends. And on any given day, we had 5 minutes of measurement with patch IP, followed by 5 minutes of measurement with the transthoracic IP. This is an example of how the raw patch IP data could look like, and we see the multi-frequency component there. We measure with 4 excitation frequencies, 550, 100, 150 kilohertz. And after obtaining the signals, we pre-processed them with our published pipeline and extracted 3 respiratory timing markers: the inspiratory time, the expiratory time, and the respiratory rate, which is derived from this. And what did we find? Well, by comparing the feature trends for all of the respiratory timing markers across all 4 excitation frequencies, we see that they capture very similar trends. We see the inspiratory time first showing an increasing trend, the expiratory time showing also an increasing trend overall, but there's one of the excitation frequencies that was different or a little bit different, and then corresponding respiration rates from admission to discharge. Now, while there are really no common known normal values for inspiration time and expiration time, but we do know that respiratory rates of 20 are not normal. And that's what we're seeing in the last plot, and that was expected given the patient population with heart failure with signs and symptoms of respiratory distress. And one note that I want to make here is that we can see that the multi-frequency IP analysis, although just shown here to show how each of them track the changes, they could be used to increase robustness as we could average them out, or as done in recent work, we could pick the excitation frequency with the highest quality to compute the features. And before moving, I want to point out that we did expect differences on the measurements at any given day, as for example, for expiration time, because of the asynchronous nature of the recordings, where we put one place configuration first and then the other. Another interesting result was the finding of this abnormal respiratory pattern, that's called the Cheyne-Stokes respiration. And on several of the patients, and in particular for this one, on both admission and discharge, we can see in this plot that we have the measurement days on the rows and the configurations on the columns. And this pattern, which is very interesting, consists of this cyclic event of increasing to decreasing breathing depth bounded by these apneic periods. And the reason why it's an interesting finding is because it's a common finding in heart failure, and keep in mind that these are patients with heart failure, and it's associated with increased mortality. And in fact, this patient had an advanced stage heart failure and had a very poor prognosis according to the clinical notes. And it is nice to see that both configurations are capturing very similar information, even though they are being placed at different times. And they are actually virtually capturing the same number of CSR events. And we also found this abnormal respiratory pattern on 3 of the 7 patients, which was interesting given that all of them were heart failure patients. All right, so to provide some concluding remarks, this work has presented an initial validation of the patch IP configuration against the conventional one in a clinical setting. The more convenient patch IP configuration demonstrated that it can capture high quality respiratory waveforms and track changes in breath-by-breath features, as we've shown with the markers. Now that we have, in previous work, validated this patch IP configuration in static and active scenarios in a controlled environment in a healthy population, this work is now presenting an initial validation in a clinical setting in a patient population. So the future work for this patch IP configuration could very well be further validation studies in larger populations. It was only N equals 7 and N equals 5 for 2-day measurements. Different diseases, not only heart failure, could be different things, and also in other uncontrolled environments, such as perhaps other clinical settings, at home and active scenarios. And that's that's it for today. Thank you so much. I'll be happy to answer questions now.